Hi, good morning, everybody. This is exciting. That's our last Vanbug seminar of the season. I'm very happy that we've managed to pull it through despite like everything that has been happening this year. So today I'm quite excited to talk about, introduce our speakers, and we also have one big event coming up next month. So this is going to be our program for today. We're going to start off with a bit of announcements. We're already a few minutes late, but that's usually how it goes. And then we're going to have an introductory student talk, followed by our main talk by Dr. Carl. And then there will be more or less open until 12:30. And then the big event that I want to tell everybody about is that we're organizing next month. So we usually don't have a Vanbug on in May, but this year we decided that we're going to have a special event. So this is going to be our first in-person event since February 2020. So it's going to be a career panel event at Langara College, our sponsor, and it's going to be back to our old time on Thursday between 6 p.m. and 8:30 p.m. Check our websites. The details for the event are being finalized. There will be food, there will be beverages, and it will be a great opportunity to finally see you again in person. And the other thing that I want to tell you about is we are preparing for the next season. Hopefully, the plans are going to be for it to be back in person. And we would love to hear from you about what speakers would you like to see. So if you have any keynote speakers that you have in mind that you think would be a great fit for Vanbug, please go to the website, and on the main page, you'll see here a suggest the speaker box. Click on it and fill the form and let us know who you think would be a great fit for us, because we are looking for speakers. And with that, I want to just acknowledge our sponsors, without whom Vanbug would not be possible. So that includes the Graduate Studies in Bioinformatics program between UBC and SFU, Genome BC, Ecoscope, the Canadian Bioinformatics Workshop, Connectia Health, Langara College, who is hosting us next month, Stem Cell Technologies, and also Vanbug is affiliated with these organizations, ISCB, and two bug sister organizations, Montreal and Toronto, Monbug and Torbug. So with that, I'll just quickly introduce our speakers today. So first, we have Bradley Jones. He's a PhD student in bioinformatics in the Joy Lab, and he'll be talking to us about evolutionary modeling of HIV persistent reservoirs. And our main speaker today is Dr. Carl. So Dr. Carl is an assistant professor at the School of Biomedical Engineering, and his work involves looking at large amounts of data to understand the gene expression patterns and the gene regulatory and gene regulation as well. I don't want to take too much of our speaker's time, so let's start with Bradley. Brad, please go ahead and share your presentation. Brad, I think you're still muted.
Yeah. Hi, so I'm Brad Jones. I'm a research assistant at the BC Center for Excellence in HIV/AIDS, and I'm a PhD candidate in the Joy Lab at University of British Columbia. Today I'm going to talk about my thesis research, which is evolutionary modeling of the HIV persistent reservoir. So human immunodeficiency virus, or HIV, is a blood-borne retrovirus which causes acquired immune deficiency syndrome, or AIDS, and it's responsible for an ongoing pandemic with more than 30 million people currently affected and 1.5 million new infections just in 2020. So currently, HIV infection can be managed with a combined antiretroviral therapy, or CART, but this therapy does not offer an HIV cure, which we shall see why soon. So throughout the course of HIV infection, HIV integrates its genome into host cells, and then these cells will go on to later produce virus, and the cell will then perish. Some of these cells will, instead of producing virus immediately, will go latent, and then they will persist until later reactivating to produce virus, continuing the infection. So antiretroviral therapy is really good. I mean, doesn't interact with this latent reservoir. It just stops the virus from replicating. So when you're on therapy, even though your viral load will go down to undetectable levels and you will no longer be infectious or progress to AIDS, if you discontinue therapy, the virus will start replicating again, and you will go back to the same viral load as when you initiated therapy, and it'll be like you never went on therapy at all. So to come up with a viable HIV cure, we need to entirely manage this latent reservoir, and to be able to do that, we need to learn more about the reservoir. So in particular, the thing that I'm interested in is when the latent proviruses are integrated and how long they persist. So HIV has a very high mutation rate, and it will rapidly evolve and adapt to the host throughout the course of infection. And at the same time, viruses will be integrated into the persistent reservoir where evolution will cease. So if we sample sequences during active infection from plasma, and then and sample sequences from the reservoir, then by comparing these sequences, we can infer, comparing the latent sequences to the active sequences, we can infer the integration dates of the reservoir sequences. And to compare these sequences, we use something called phylogenetics. And phylogenetics is a study of phylogenetic trees, which encapsulate the evolutionary relationships between organisms or sequences. And basically, the path lengths on this phylogenetic tree encode the evolutionary distances between the different sequences. So I will be using three different techniques in order to estimate these integration dates. The first is phylogenetics and linear regression. The second is phylogenetics with Bayesian analysis, and the third is using simulation. So to estimate integration dates with linear regressions, we start with plasma sequences sampled longitudinally from the reservoir before therapy is initiated, and also sequences from the reservoir collected from blood during suppressive therapy. Then we infer maximum likelihood phylogeny from these sequences, and we route the tree with root-to-tip regression to maximize the correlation between the collection time of the plasma sequences and their divergence from the root. Then we fit a linear regression relating the collection date and the divergence of the plasma sequences, and using the divergence of the, and finally using the divergence of the latent sequences, we can, and plugging it into the linear regression, we can come up with an estimate of their integration dates. So if we apply this to real data, we can see that the integration dates are spread throughout the course of infection. And this implies that the HIV reservoir is, at least in this individual, is genetically and chronologically diverse, which makes it more challenging, which potentially makes it more challenging to develop a cure for. But this method is, so the linear regression method of estimating integration dates has a few is a little bit simplistic, and in particular, it relies on the strict molecular clock hypothesis, where mutations will arise linearly over time, which is not entirely realistic. So I wanted to investigate more sophisticated methods to estimate these integration dates, and the technique that I'm investigated next was using Bayesian methods. So in particular, using Markov chain Monte Carlo to sample phylogenetic trees using the software Beast2, and I developed this Java package that I call BBD to work with Beast2 to estimate HIV integration dates from the sample of trees. And basically, if you have basically you start with a prior distribution on the dates of the integration that you kind of think that they might have, and then by running the MCMC, you end up with posterior posterior sample on the integration dates. So if we apply this the Beast2 Bayesian methodology to the same participant data that we had before, with the longitudinal plasma sequences collected before therapy and the reservoir sequences collected after therapy, we get similar results where the dates are spread throughout the course of infection. So in this figure, the is a tree where the distances are in time, and the black diamonds correspond to the latent sequences, and their x-axis position is their estimated date. The pink bars show the 95% highest posterior density. And the main thing to note about this data is that in this data set, 2 of the sequences date very early, before the earliest plasma samples were sequenced, and this was not seen with the linear regression. So these methods are all well and good, but we don't actually, we keep just using empirical data, we can't tell how accurate these methods actually are. And so to evaluate how accurate these methods are, it would be nice to know when the viruses are actually integrated into the reservoir. And one way we can do this is with simulated data. So my next thing I'll talk about is how I modified this software SantaSim, which will take a population of sequences and evolve them over time. And I modified the software to allow multiple compartments. And so that, because it could only simulate a single population of sequences with homogeneous characteristics. And then I used my modified SantaSim to develop a simulation of the within host HIV evolution, including the persistent reservoir. And then with the simulated data, I then used that to compare different date estimation methods. So using 100 simulated data sets, I compared 3 different dating methods. So the linear regression, which I've already discussed, and then 2 other methods that were developed previously. So the first one is the closest sequence method, where you build a phylogenetic tree of all the sequences, and then you compare and then you find the closest sequence from the plasma that's to your latent sequence that you want to date, and you just give the plasma sequence that date of the plasma sequence, and then LSD assigns dates to the internal nodes of the tree and the latent sequences to minimize the least squared difference between the time in the tree and the genetic distance. And then I also compared the Beast2 to the linear regression and the LSD on just one simulated data set, because the Beast2 method is a little bit more involved and takes a little bit more time to run the simulations through. And I found that where before the LSD was the most accurate method, with the beast2 later ended up was shown to be the most accurate, being having half the RMSE of the LSD method. So in summary, the closest sequence is the least accurate, and the beast2 is the most. And then maybe not that surprisingly, these methods are ordered by their accuracy here, but they're also, in some sense, this is also the order of their complexity, with the closest sequence being the simplest model and the beast2 being the most complex. So in summary, HIV proviruses are seeded continuously and persist regardless of which the methods you use to date the sequences, and then more sophisticated methods are more off accurate, and understanding the dynamics of HIV persistent reservoir is important for HIV research. And with that, I'd like to thank the co-authors on my work and the members of the Joy Lab and the BC Center for Excellence in HIV laboratory. I'd like to thank my funding sources, which are CIHR and NIH, and I'd like to send a special thanks to the study participants, without whom this research would not be possible. Thank you for listening, and I'm happy to take questions at this time.
Thank you so much, Brad. That was really interesting. And if there are any questions, please post them in the chat, or if you want to speak up, just raise your hand, and then you may speak up. I guess I'll start with a question. So in this context, is it like when we were describing a reservoir, you're describing within like a single patient?
Yeah, sorry, yes, sorry. Yeah,
and
yes, it's within a single patient.
But then, like in these cases, like so like are these patients not on therapy for extended periods? That's why there is an evolution in their HIV sequences, or not?
Yes. So yes. So if you're on therapy, typically there will be no evolution.
Mm-hmm.
Unless you have a resistant virus, then it's a different story. But
yeah. And are these like sampled at like the actual like sequencing data? Is it like sampled at multiple time points, or is it just at the end time?
They're sampled at multiple times and at the end time, yes. Mm. So the sequences from what I was calling active infection, so not having without therapy, those are sampled over multiple periods of time, and those samples were taken from in HIV care, it's standard to take viral load testing every so often, and these samples were taken from the leftovers of that test.
Mm-hmm. I see. Cool. Well, thank you so much. There's a question in the chat, so I'll read it out. What would you say is the next step for your work? Was your goal mainly to compare methods here, or are you aiming to make further biological inferences from what you have found?
So actually here, I actually did not show all of my work. I just tried to highlight the main projects that I've been working on, but I've also like applied some of these methods to like other patients and other in HIV, to other patients. And yeah, my goal is basically just to apply this to multiple and try to make general inferences, which is not easy, because pretty much every single person that's infected with HIV more or less has their own the virus has its own trajectory in how the evolution happens within that person, and they all have different interactions with the therapy that they take and etc. But yeah, my main thing is to use this on more patients, and to maybe explore other models too.
Awesome. Well, thank you so much, Brad. I guess if there are more questions, people can take them offline or maybe towards the very end. And thank you so much, Brad. I guess I'll hand it over now to Dr. Carl. For the audience, if you do have questions, please feel free to post them at any point throughout the talk. I guess the talk is going to be like more or less in like covering different themes or different topics. So questions throughout are encouraged, and definitely also at the end. So with that, please take it over, Dr. Carl.
Can you see my screen?
Yes.
Great. Thanks for confirming, and thank you for the introduction. And thanks to Brad for introducing some of the evolutionary concepts that we'll talk about in the later part of the talk. So today I'm going to talk about a very interesting big data set that gave us numerous insights into how gene regulation works, and also how gene regulatory sequences like promoters, how they evolve. So this is the overview of my talk. First, we're going to talk about the particular problem we're trying to solve, predicting expression from sequence. Then we'll talk about how we generated data, specifically about 100 million random promoters, to learn how to predict expression from sequence. Then we'll talk about two different models that were used for very different purposes. A biochemical model that we were using to infer how gene regulation actually works, and a deep learning model where we used it for scaled inference to study the evolution of regulatory sequences. So one reason we should care about how gene regulatory sequences encode for expression levels is that a large amount of the genetic variation that is implicated in common complex inherited diseases, things like autoimmunity and heart disease, they lie in gene regulatory regions. So that's shown in this particular paper here. There's actually quite a number. Pretty much every time a new genome-wide association study is done, they do this kind of analysis and find that by and large, a lot of the polymorphisms associated with diseases are in regions that are probably gene regulatory regions. So here they're showing it in DNase 1 hypersensitivity sites. These are regions like enhancers and promoters. So these variants are somehow contributing to disease. And the model we think this works by is, so here I'm showing you have one allele and the other allele, and they are in some sort of regulatory region controlling the expression of this yellow gene here. So the way we think it works, there are these proteins called transcription factors, and they essentially can read the sequence of the DNA. So here, this particular transcription factor finds its binding site in the DNA, activates gene expression for the yellow gene. But in this allele, there's a mutation in the recognition site for that transcription factor. And so it doesn't bind effectively there and does not activate this yellow gene. And this happens in some particular cell type and affects some gene, and eventually that somehow leads to an increased risk of disease. So this is one motivation for why we want to understand this sequence to expression relationship. Because there's so many of these polymorphisms, we don't want to measure each one. We want to build a model that can actually predict the effect of these mutations. So how can we learn the language of regulatory DNA? We already know a lot about the alphabet. So DNA is comprised of these 4 bases represented by 4 letters, A, T, G, and C. The words in regulatory DNA, these correspond to things like transcription factor binding sites, as shown, as represented by these motif logos. The grammar, we know a lot about the grammar through specific biochemical studies, but by and large, we don't know most of the parameters. We just know certain ways in which they can work. So for instance, transcription factors can modify expression directly. They can also act by modifying chromatin. So here it's inhibiting nucleosome formation. They can also modify each other, either preventing other transcription factors from binding through competition or promoting the binding of other transcription factors through cooperativity. So just to give you an idea for how complex this problem is, we're just going to consider one of the parameters we would need to learn, and this is cooperativity between transcription factors. So any pair of transcription factors can potentially interact. We haven't tested all of them, and so we would have to learn this. And there are the number of transcription factors squared possible interactions between pairs of transcription factors. They can interact at specific distances. So for instance, it might interact at an offset of 8 base pairs, but not at 7 and not at 9. So from what we've seen, you have to consider about 32 base pairs offset between the two transcription factors. It could be one or more of the positions within 32 base pairs offset. They also have an orientation. So here I'm showing the 4 possible orientations. They can be arranged in different ways, and it's possible that the interaction happens in only one of the 4 different interactions. So that's number of transcription factors squared times the distance between them times the number of possible orientations of the motifs. So just to give you an idea of what this looks like, humans have about 1,640 transcription factors. So only considering cooperativity, we now already have over 300 million parameters that we have to learn. And if we're just studying the regulatory regions of the genome, each cell has on the order of about 50 to 200,000 different regulatory regions. So clearly the number of parameters here that we want to learn is vastly outnumbered it vastly outnumbers the number of regulatory sequences we would have to learn these parameters. So we're going to take a step back and use yeast as a model system. And one of the benefits of using yeast is that it has far fewer transcription factors, about 209. And that means, just considering this cooperativity interaction, again, there's only now about 500 million parameters to learn. Unfortunately, you also have many fewer regulatory regions, only about 6,000. So in either case, you have far too few examples of regulatory DNA to learn the parameters that you want to learn. So now we're going to talk about the data set that we created to learn this complex problem. So we took a step back and basically thought, what is the ideal data set for learning this sequence to expression relationship? So ideally, they'd be high quality expression measurements so that there's lots of signal and very little noise, measured in a controlled system so that there aren't other factors that are affecting it that you're not considering. The sequences should be unrelated, so things like training test violations, where there's a very similar sequence in training and test, are avoided. Diverse transcription factor binding sites, so that you can learn how each transcription factor is working, and many, many examples. We know we'll need a very large number of examples to learn all the complex parameters. So one solution that people had tried was to use native sequences. So like taking the promoters out of the yeast genome and testing those. But this is not a great approach because the sequences are often related to each other through homology. You don't have that many diverse transcription factor binding sites. It's just whatever happened to be in the genome, and you don't have many examples. Like I said, there's only about 6,000 promoters in yeast. So people have also tried design sequences, and these are great because you can make the sequences unrelated if you want. You can put in whatever transcription factor binding sites you want, but you can't generate that many examples. So right now, Twist offers an oligo array of about 700,000 sequences, which sounds like a lot, but it's going to cost you a heck of a lot of money, and 700,000 is still not enough sequences. And to get another 700,000, you have to pay twice as much money. So it's also not an ideal solution. So we considered whether we could just use random DNA. And it seems to be great because the sequences are, by definition, unrelated. If you're sampling random sequences from the vast space of possibilities, the odds that 2 sequences are going to be related significantly is very, very low. You can also generate many examples, and this is basically because of the way DNA synthesis works. You can buy for $100 a tube containing trillions of distinct random DNA sequences. But what about transcription factor binding sites? do random sequences contain transcription factor binding sites? So we did an analysis of the information content of yeast transcription factor motifs, and actually we updated this recently with human transcription factors, and it shows pretty much the same thing. So you can see the expected frequency in random DNA on the x-axis, and the cumulative fraction and also the density are shown in these black and purple curves. So for instance, this Reb1 motif, this is a transcription factor in yeast. This one has a relatively high information content, but even Reb1 has binding sites in random DNA. You expect to see them about once every 16,000 base pairs. So most transcription factors are more like RISC-3, have a intermediate information content, and here you expect to see them about every 100 base pairs or so, so quite frequently in random DNA. So actually you can calculate the expected number of binding sites in a random 80 base pair sequence, and we used 80 base pairs, so that's why I'm telling you 80 base pairs. And you expect to see about 138 binding sites for about 68 different transcription factors. So a very large number of binding sites. So but this is just what information theory is predicting. Let's check to see what actually happens when you test the expression level of random sequences. So here we cloned in a random 80 base pair sequence, and we put it in the context of this promoter scaffold. And this is basically sequences that look kind of like the surrounding regions of yeast promoters. And we have this YFP reporter gene. And here I'm showing these 5 curves. Each of these represents a single clone. So many yeast cells were measured independently, but they all had the exact same promoter sequence in them. And you can see that you get 5 distinct expression levels, 1 per clone. This pink curve here represents the entire library. So rather than 1 clone, we now have a very large number of clones all mixed together. And so you can see the extreme diversity in expression. And the only really way this can possibly happen is if these sequences all contain different binding sites for transcription factors. So each are encoding their own unique expression level based on the random assortment of binding sites it happened to get in the random sequence. So solution 3 looked pretty good, random DNA, so we went forward with that. And now we needed a way to get high quality expression measurements in a controlled system and a very large number of these measurements. So here's how we did it. So again, this is the random 80 base pairs in the same context as before. And now we're going to sort the cells into different bins of expression. So here we have the lowest expression and the highest expression. And we sorted those cells, we regrew the cells, and we did flow cytometry to see what the expression levels of those cells were. So you can see we sorted cells into bin 18 and we sort them, we flow them again. And sure enough, bin 18 has the highest expression level out of all of these. And you get this nice ladder corresponding to the bins that we sorted the yeast into. So now we have the different promoters that had different expression levels and different tubes. So all we need to do now is just isolate the promoter DNA and sequence it. And this gives us this really large table of expression levels in one column and the DNA sequence that encoded it for the other column, where the expression level is just the bin we found the promoter in. So like bin 18 is the high expression bin. So if we saw this sequence in bin 18, we say it has an expression level of 18. And the DNA sequence is just what we read off of the sequencer. Remember, they're random DNA sequences, so we don't have a reference to compare to. All we have are the reads that we saw from the sequencing run. So using this approach, we can generate about 30 million sequences per experiment, which is quite a few. And to date, we've quantified over 15 gigabases of new regulatory DNA. This is in yeast. So we now have several human genomes of promoter sequence in yeast. So a lot of data to learn this function relating DNA sequence to gene expression. So that's the data. So next we're going to talk about the 2 models, starting with the biochemical model. So we called it variously the biochemical or the interpretable model, but it's interpretable in that the parameters that it's learning are explicitly trying to capture how transcription factors work. It has pretty good predictive power, but its main utility is that it's interpretable. You can look at the parameters and see, like, this particular parameter represents some feature of a transcription factor, so you can directly interpret them. This was published in Nature Biotechnology in 2020. And the neural network that we'll get to later was also recently published. So we're talking about the interpretable model first. What does it learn? So it learns these transcription factor motifs that capture what the transcription factors like to bind. We actually initialize them to known motifs for yeast transcription factors so that it starts out knowing roughly what each transcription factor prefers to bind to. But then we allow the model to update these so that it can actually better represent the specificities of the transcription factors. It also has to learn their ability to modify chromatin. So we call these potentiations because the binding site for one factor can potentiate the activity of another. For instance, that second factor might only work in the presence of the first, and this can act through chromatin. We also learn how each transcription factor affects expression directly. So does it increase expression? Does it decrease expression? That would correspond to activators and repressors. So we'll step through how this model actually works. And this is actually real data here. So what you're going to see is an actual run through for a specific promoter sequence using one of the models that we made. So we have our motifs as part of the model, and we have the promoter sequence that we're trying to predict the expression of. So we essentially scan the promoter sequence with each motif. This is equivalently a convolutional layer in a neural network, or it's just scanning with a position weight matrix. They're both equivalent operations. And what this gives you is for every transcription factor, you scanned both DNA strands for that same DNA sequence. So you scan it in the forward orientation and scan it in the reverse orientation. And for every position, so you get these predicted binding for each transcription factor at each possible location. Then we summarize this basically for transcription factor 1. We add up all the binding across the entire sequence so that we get a summary for how much this sequence is bound by each transcription factor. One value per transcription factor. And the model has learned parameters here. We call them the potentiation parameters for how much each transcription factor opens or closes chromatin. So for instance, these red bars here represent transcription factors that open chromatin. They have a high potentiation weight. And through this, we basically run it through a logistic function to get a latent variable here, the predicted accessibility of the sequence, which goes between 1 for it's 100% accessible, or 0, it is completely covered by nucleosome, so the sequence is inaccessible. Okay, so we then actually use this accessibility value to reweight the DNA binding values. So essentially, the idea here is that if it is 100% accessible, then you're just multiplying this by 1. And so the binding, the predicted binding of the DNA in the absence of chromatin is the same as in the presence of chromatin because it's completely accessible. There's no change. But if the accessibility were 0, so 100% of the time there's a nucleosome there and transcription factors can't bind at all, this would be this vector here multiplied by 0. And so no transcription factors would bind anywhere in the sequence because instead you have a nucleosome covering that sequence. And then it's just the dot product here between the predicted chromatin binding and the activity that is also learned for each transcription factor. And that is the predicted expression level. So this part is just a linear model. transcription factor binding times the activity of each transcription factor plus some bias term. And this is like a logistic model. And the beginning part, predicting transcription factor binding, it's a little more complicated, but it's actually not that complicated either. It's just position weight matrix scanning, and you get probability of binding at each position. So it's a relatively simple model and it works pretty well. We extended this model to be a little more complicated, and I'll explain how now. So the idea here is that these transcription factors, they can actually do different things depending on where they're bound in the promoter. And that is because they can interact with things that are nearby. So here I'm showing this pink transcription factor as it moves across. Its goal basically is to interact with this pink part of the pre-initiation complex, and it can only do so efficiently when it's close by. So it would have a high activity over on the right-hand side and a low activity on the left-hand side. So we included parameters that capture this. So there's a parameter for each DNA strand, for each transcription factor, and for the 110 different locations of the promoter. So a total of 55,000 parameters. And that's, this is the modified model now. So everything up until predicting the accessibility is exactly the same as the model we just talked about. But now instead of scaling this summary of transcription factor binding, the total amount of binding across the sequence, we're doing it in a position specific way. So we're taking this tensor here and multiplying that by the accessibility value, scaling it by the accessibility. And now instead of having one activity per transcription factor, we have 2 times 110 per transcription factor. So one for each location and orientation. And then you just do a element-wise multiply here to get the activity of each binding site. And then basically you just add that all up. Again, it's like a linear model at this point. So this interpretable model works pretty well. It's got Pearson R squared in random DNA. So this is the predicted expression level versus the measured expression level. And these are for random DNA sequences. It can explain about 94% of the variation expression here, which is quite good. It's a little lower for native sequences. So here we've taken 80 base pair chunks of actual yeast promoters, stuck them into our reporter system and measured them, their activity there. And we can explain just over 88% of the variation in expression here. So yeah, the biochemical model works pretty well. So let's see what it learned. So the models are interpretable. So we can just ask them how these transcription factors are working. So I'm going to go through really fast here, but one of the things it learned is condition-specific regulators. So I didn't tell you, but we actually did these experiments in different conditions: glucose, galactose, glycerol, and we've since done more. So we found that, of course, the GAL4 transcription factor, which is known to be galactose specific, is specific to galactose. No surprise there. So that's like a positive control. We found some other regulators that are specific to glycerol, and these are mostly new. We were able to predict chromatin structures. So this just shows the predicted and measured accessibility and also nucleosome sequences across yeast promoters, just an average of all of them. It's a metagene profile there. We compared the learned and initial specificities of transcription factors and found that by and large, the motifs that the model had learned were actually better than the motifs it started with at predicting other types of data like ChIP-seq. And it was able to predict how transcription factors how their activity is affected by position, and we'll get into this in the next slide. So here is one example. So this red curve here is one example of a particular transcription factor for one strand, what its model learned activity across the promoter was. And you can see here that it has this periodic structure to it. And this corresponds exactly to the 10.5 base pair helical turn of DNA. So the idea here is that there are transcription factors that, like, as you're moving down the DNA, you're also actually going around the DNA. So transcription factor that, so here the pink one has to interact with this pink domain. It can only do so efficiently on one DNA helical face. So as you're moving, every 10.5 base pairs, you're back on the same helical face, thereby resulting in this periodic structure. And we actually showed this for a very large number of transcription factors. So here's another example. This is a motif for a transcription factor called MGA1. This is the model predicted, model learned parameters for how this MGA1 motif affects the expression of the reporter gene at every possible position for the top and bottom strands. And so we did an experiment. If you're gonna zone out for the rest of the talk, this part is, I would say, the most important, because it'll really convince you about the abundant transcription factor binding sites in random DNA. So we did this experiment to validate these predictions. So we have the predicted activity of this MGA1 motif for top and bottom strands. And what we did was we inserted the MGA1 motif at every possible position in one strand, in the background of a random DNA sequence, and in the other strand. And we measured the expression level. So it was surprising to some, but not to me, because I already knew what the model was going to do. The expression level actually does not correspond to the parameters learned by the model, not even slightly. And there's a very interesting reason for this. The model is actually predicting this. So before we had the experimental data, I already saw the results, and I knew this experiment would fail before even doing the experiment, but it was still a very interesting experiment, so we did it anyway. You can see the predictions very closely match the measured expression levels for both motifs on the top strand and motifs on the bottom strand, but they don't correspond to this periodic structure. And you can ask this model what's going on. So we did that. And it turns out that as you're tiling this MGA1 motif across the sequence, you are creating and destroying many other transcription factor binding sites for both strands. And that's shown here. So these are motifs that affect expression directly, and these are motifs that affect the accessibility of the sequence. So as you're tiling this motif, you are perturbing many other binding sites that were there already, and you're also creating new ones at different locations. So just how many transcription factors regulate a given sequence? We can actually computationally delete these transcription factors. It's a model, and we can perturb the model. So we computationally delete a transcription factor and basically get an interaction effect size for how much this particular transcription factor influences the expression level of that particular sequence. We can do it for one transcription factor. We can do it for many transcription factors for any given promoter sequence. So we did this for all yeast promoters. Here are two of them. So these are interesting because they have very different regulatory logic, but they encode for the same expression level. So here we have one, this is a promoter sequence. The transcription factors are all these nodes on the outside of these graphs. So this particular sequence has one transcription factor that has a really big influence on the expression of this sequence. You knock out this transcription factor or you delete its binding sites in that gene, and expression will change very substantially. Now this sequence has the same expression level, but really no transcription factor makes a very large impact on the expression of it. This one is the biggest impact, but even this one's a minor, it's like a drop in the bucket relative to the many other small effect sizes affecting the expression of this sequence. So we can look at this across all sequences. And so here I've created a cumulative distribution function for both the amount of expression explained and the strength of the regulatory interactions. The interaction strength is on the x-axis. And the best way to understand this is to just go through this example. So if we just look at twofold interactions, we can see that about 1 in 700, so like 0.1% or so of these regulatory interactions are greater than twofold. Meanwhile, 94% of the expression has already been explained by interactions that are less than twofold, leaving 6% explained by the twofold or greater. So it's like 0.1% of interactions explaining 6%. So they do explain a disproportionate amount of the expression, but 94% of it is less than twofold expression interactions. So conclusions for this part, the biochemical model. So transcription factor binding sites are abundant in random DNA. We can learn how transcription factors work from sequence expression measurements alone. And these regulatory networks are far more complex than previously appreciated, including a very large number of low effect interactions. And if there are any questions about this part, now is a good time for that.
I guess I'll ask a question in anticipation of other people's questions if they have any. So when you're building this large data set, I mean, do you think it is a disadvantage that you're really only isolating the end, like you're only looking at the effect of the nucleotide sequence, and there's no like other effects that could be like non-sequence effects? For example, like if there's epigenetic effects that are contributing that you would not be able to capture because you're like you've isolated all of this. Do you think that is important or relevant in this for your models here?
I think it is important, but I don't think it's relevant to these models because we're explicitly trying to rule out those effects here. So I totally agree that those effects are important and will affect things, but the way I see it, it's like a separate knob in the regulation of gene expression. And if we try to study both at the same time, then we might not be able to solve either. So we're trying to like isolate our variables here.
So like this, I guess, box for generating data sets, its applicability to different species or different genomes, it is only a question of like changing the set of like the promoters that you care to look at, the regions that you care to look at, but not like the transcription factor random sequences.
Yeah, so we haven't looked comprehensively across all species, and actually bacteria are interesting because they seem to actually, they do have activity of random DNA, promoter activity, but it's actually less than eukaryotic DNA. But it looks like humans too, this approach will work. It's just an engineering challenge to get it to work, and we're actually doing that now.
We have a couple of questions. So first from Annie, how does your model work for pioneer factors that in theory can bind to closed chromatin? Yeah,
so that initial step is predicting binding, assuming that that chromatin is open. So the pioneer factors would presumably have binding sites that it could bind to if it's accessible. And if they're binding there, they will get a high potentiation weight, which basically tells the model that if there's a binding site for a pioneer factor, chromatin will be open. So it's kind of learned indirectly in the model. We just don't call it pioneer activity. Part of this is historical. They didn't call them pioneer factors in yeast, but that's essentially what potentiation is learning.
Another question from Maxwell. Very interesting project. How much do you think the model depends on the reporter gene and the sequence surrounding the random part?
If you look at the paper, we showed that the surrounding sequence does affect the position-specific activities. So that definitely is an important consideration. The reporter gene itself, to some degree, it probably does, but I would hope that not very much. We haven't comprehensively looked at that. From what other people have shown, it probably doesn't make much of a difference because it's just a little bit too far away.
Cool, thank you. We do have another question. The model sounds like bad news for literature saying simple models of transcription factors regulate other downstream transcription factors. For example, BCD activates HB, etc. Will this undermine the way we think about existing regulatory models? For example, the Eve enhancer is very well studied. Is it still interpretable the same way?
Yeah, that's a complex question, but the short answer is it has really changed the way I think about these gene regulatory networks. And the way I think of it now is basically the null hypothesis at this point is that it's a fully connected network. Everything is regulating everything, it's just the effect size. And historically, what we've been studying are these large effect sizes. And so you like mutate a TF binding site at the Eve enhancer, and you can see like this hunchback binding site was super important. You have a huge effect on expression when you mutate it. You're not going to see the effects that are small. So you can—
Would that like, so in this view is also like in a sense compatible with the like all of these, like when you see like the snip effects, like you see like so many SNPs could be affecting like a specific phenotype, because like would that like maybe is because like it is affecting so many different transcription factor binding, but each of them is just a little bit. Could that like be like in the same view?
Yeah, it definitely could be one explanation for that.
So we have one more question. For the many non-coding GWAS loci that don't associate with changes in expression, how useful do you consider this model? I guess it's a bit similar to what I was just saying.
Well, yeah, so I don't know. It's important to think about this in the background, but of course, many GWAS signals will not act in this through a gene regulatory mechanism. So that's something important to consider too. Also, with what I talked about before was in yeast. So we still have to show the same thing in human, but honestly, it's like people arguing that yeast are more complex than humans. I don't really think that's going to be the case, but it remains to be seen.
Okay, so I guess let's take this last question before and then move on with the presentation.
Okay.
So the last question from Oriol, great work. We discussed it last week during our group meeting that we're developing an explainable deep learning architecture that is linear. It does not learn nonlinear interactions such as cooperativity. And performs well as more complex models such as Basse or Dankyu. In your opinion, what is the percentage of the total contribution to gene regulation by cooperativity and similar complex effects?
Yeah, that's also a difficult question to answer. So these are the kind of events that you expect to be very rare in random DNA. And if they're rare, that means that if you just look at the overall Pearson correlation, basically it makes no difference if you've captured them or not. To the sequences that are affected by those interactions, it is probably going to matter a lot. So people have studied, yeah, like the Eve enhancers and other cases where we know that these things are happening and they are very important, but they do seem to be the minority of enhancers.
Cool. So I guess we'll catch up with the other questions maybe later towards the end of the talk.
Yep.
So go ahead with it.
Thanks. All right, so now we're going to talk about this deep learning model and how we used it to study evolution. So this work was done by Ishit Vaishnav, who is a PhD student in Aviv Regev's group. So I did my postdoc there. And so we started this project together while I was in the lab. He did most of the work I'm going to talk about now. Yeah, so he made this neural network. It was really good, difficult to interpret, though not impossible, but we didn't try because we were instead going to use it for something else. So this just shows the accuracy of the model. It's better than the interpretable model by about 2% here and 3% here. If all we wanted here was something that predicted expression really well, it didn't make sense to use an inferior model, so we used Isha's deep learning model. And I'm not going to go over the architecture at all. It's in the paper if you're interested. We're just going to talk about using it as an oracle to predict the effects of gene expression. So we had our data that I talked about before, trained a deep learning model that predicts expression from sequence, and now we're going to use it to study the evolution of regulatory sequences. So this paper is part of a group of papers that are trying to predict the effects of mutations on the function of either regulatory or protein sequences. These are increasing as we have gained more power to understand how these systems work. So keep in mind that this is part of a greater literature of doing similar things. We did a lot more of the evolution stuff in the regulatory sequence space. So we're going to start out by simulating neutral drift of regulatory sequences. So say you have the starting sequence and it's going to accumulate mutations over time. There's no selection happening here. So we're considering all possible mutations, and then you just pick one at random and you keep adding mutations until you get n random mutations added to the sequence, and then we're just calculating using the model as this oracle, the predicted change in expression. We predict the beginning, we predict the end, we calculate the difference, and we see, plot the difference on the y-axis here relative to the number of mutations added to the original sequence. Of course, 0 mutations, the ending sequence is the same, so that's why there's no change. And you can see that it diffuses fairly rapidly, and by about 32 mutations added, now this is an 80 base pair sequence we're considering, so 32 out of 80 base pairs mutated, you're almost as different in expression as if you were two completely unrelated sequences. So it can diffuse fairly rapidly. So we did an experiment to verify this, and here we're showing measured changes in expression in the light gray and predicted in dark gray for 1 to 3 mutations, and you can see that the measured and predicted closely match. More generally, these mutations, so these are single base mutations, they seem to follow a power law where about 10% of the mutations explain about 50% of the expression change. But the other thing I want you to remember here is that there isn't really a line at which you can say the SNP, the single nucleotide mutation, is not going to change expression. They're all changing expression to varying degrees until you get to really trivial changes in expression. So this also makes sense given the last part I talked about, how there are these transcription factor binding sites all over even random sequences. So you put mutations anywhere, it's going to be either creating or destroying a bunch of weak binding sites for transcription factors you didn't even know bound that sequence. So next, we're going to use the same kind of paradigm. You're using this model to predict how quickly strong directional selection can operate on a regulatory sequence. So we start with some sequence and we generate all possible mutations, and now we're going to select the best mutation. So that's either the highest expression or the lowest expression. And we do this n sequential times until we have created this trajectory of ever increasing or ever decreasing expression. And you can see that the predicted expression here, it gets close to saturation after like 3 or 4 mutations in either direction. So it can actually modify, selection can modify the expression of these regulatory sequences quite rapidly. And this is the experiment showing that this is true. So we synthesize these sequences and measure their expression. And you can see again, 3 or 4 mutations is pretty close to the endpoint. It's already gotten most of the way to the expression extremes. Now, we redid the same thing, but this time doing the difference in expression between 2 media. So defined minus complex media in this case. And actually here, the process is much slower. So optimizing in 1 condition is fast. Optimizing in 2 conditions where you're trying to get opposite expression extremes in the 2 different conditions is much harder. So you can see that here. So just tracing this red curve where we're maximizing this difference in expression, you can see that even after 10 mutations, It's gotten close to the bottom, but it hasn't gotten close to the top yet. So much slower for competing objectives. So next, we're going to take a look at regulatory variation. So we took a look at whole genome sequences from 1,000 S. cerevisiae isolates from this paper. And what we did was we take these isolates, we identify all the orthologous promoters for one particular gene. So we have this set of natural variation in this promoter sequence. And then we computationally create a matched set of simulated variation where we just place the mutations essentially randomly. We're just matching the same numbers of mutations. And so there's no selection operating on the simulated variation. And then we just use the model to predict expression for these sequences. So we get two distributions, expression distribution for simulated and for the actual natural variation. And we calculate this, we call the expression conservation coefficient. It's just a ratio of the standard deviations here between these two distributions, the log ratio. And this is supposed to capture how constrained the sequence is in terms of its gene expression level. So we'll just show the values here. So this is the distribution of all ECC values for all yeast genes. This is the proportion of the genes landing in each bin. You can see that most of them are in the side. This is the side that implies that expression is more conserved. Left-hand side, the sequence is divergent in its expression level. So this is kind of expected, right? Most genes have a conserved expression level. Now, looking at the extremes, we saw that the genes that were most divergent, they had GO processes like carboxylic acid and alcohol metabolism enriched. And the genes that were the most conserved had these core cellular processes like RNA and protein metabolism enriched. And again, this is expected because if you mess with the ribosomes or with RNA polymerase, the cells are dead. So the only variation we see in those genes has conserved expression. So we found this interesting example, CDC36, and we found that this particular binding site for a transcription factor called UPC2, it had been mutated twice independently in evolution, resulting in the same predicted expression, low expression phenotype. So the ancestral strain had a high predicted expression, and in this lineage here, this red lineage there, it acquired a mutation. This critical C was mutated to a T, and that killed expression. In this lineage, this critical G was mutated to an A, and that separately killed expression. So the exact same binding site mutated twice independently in two different lineages for the same promoter, resulting in the same expression phenotype. So we thought this was pretty interesting. So we followed up on one particular strain and we converted this strain. It had a perturbed UPC2 binding site. And so we mutated it. It was actually mutated in two positions here. We mutated it stepwise back to the wild type UPC2 recognition motif. And for reference, this is a different strain that already had that motif and never lost it. So you can see that the predicted expression for these different promoter sequences closely matches what we measured by qPCR. And actually we found a phenotype too. So these strains, once we reconstituted the UPC2 binding site, they actually were able to grow, switch carbon sources faster than without. And you can read the paper for why we think that is. It has to do with the function of this gene. So do we have enough time for one last part? Or I'll skip to the end.
I think we have still another 10 minutes or so to go.
Okay, I'll just talk really fast. Okay, so evolvability is an evolutionary system's ability to generate new genotypes with new phenotypes. So here I'll illustrate it with this example. And it's important because it can lead to evolutionary innovation. So say we started with this sequence and over evolutionary time, a bunch of mutations resulted in new genotypes And these new genotypes also had new phenotypes. So in this system, this is a highly evolvable system because all these mutations are generating genetic and phenotypic diversity. Now, what could have happened is that selection destroyed all of these genetic variants. It did not tolerate any variation from the original genotype because those cells die. So this would be a non-evolvable system. So we can actually quantify this using the model. Basically, we consider all possible mutations to the original starting sequence and calculate the change in expression for each possible mutation. And because the order of these mutations is essentially arbitrary, we just sort this vector so that it's monotonically increasing. We call this the evolvability vector. So this captures how all the possible mutations to a sequence can alter its expression level. So we wanted to know how many types of evolvability vectors are there. So what we did, we used this framework from Smita Krishnaswamy's lab for using autoencoders to capture archetypes. So it's an autoencoder. So what we functionally do is we give it these evolvability vectors, and it's encoded into this latent space and then decoded. And the goal of the autoencoder is to have the decoded evolvability vectors closely match the actual evolvability vectors. And otherwise, it's just trying to reduce the data captured here into this low dimensional space. And how low dimensional do we want? We tested this empirically, just seeing how the reconstruction error changed for the different number of archetypes. So 3 archetypes was enough. So there are 3 different types of these evolvability vectors. And here they are. There's actually also indirectly encoded in the space a 4th evolvability archetype, the robust archetype. So the 3 actual archetypes are malleable, where many of the mutations increase or decrease expression. A local maximum, where most of the mutations do nothing, and those that do decrease expression. Local minimum is the opposite, where most of the mutations do nothing, and those that do something increase expression. So right in the middle here, we notice there's this, it wasn't officially an archetype, but it was nonetheless encoded in the space, a robust archetype, where none of the mutations did much of anything. They didn't increase or decrease expression. So we can look at different features of the space. So we're just plotting now the expression level as the color. Each dot here is a promoter sequence. And you can see that, of course, near the maximum, you have high expression. Near the minimum, you have low expression. You have intermediate expression near malleable. And at the robust cleft, you actually have both high and low expression, because you can get this this robust phenotype, whether you're stuck in high or stuck in low expression. And this is another way of showing that these are robust. This is just calculating the fraction of mutations that change expression. And you can see that very few mutations change expression here, and very many change expression on the outside. So one thing we noticed is that if you just look at a bunch of orthologous promoters, if you find one of them in the middle, you find the others in the middle. If you find one of them on the outside, the others are on the outside. So these promoters fall into, it's a spectrum really, but if it's on the outside, they can move around this space much more dynamically. If the promoter is in the middle, then subsequent mutations will keep it in the middle because it's a robust sequence. So here's just an example of each of these. So DBP7, this is a promoter. This is actually selected because it was closest to the malleable phenotype, or the malleable archetype. And you can see that orthologs of this promoter are all over the space. UTH1 was chosen because it's closest to the robust archetype. And there's actually 21 sequences here. You can't appreciate it because they're all stacked on top of each other. And this is actually in spite of the fact that there's actually more genetic diversity in the UTH1 sequences than the DBP7 sequences. So more genetic diversity, but they're stuck in this robust cleft, and evolution has fixed these in place there. So we can actually do in silico mutagenesis to further explore how these work. And here we're showing the predicted expression for each mutation for the DBP7 sequence. So this is the length of the sequence and the possible mutations. And some of these, of course, correspond to the current sequence. So we can evolve in either direction here, taking either the highest expressing mutation or the lowest in the downward direction. And you can see 2 mutations it takes you to get pretty much at the top of expression, or 2 mutations take you from this sequence to the bottom of expression. UTH1, on the other hand, it takes a very large number of mutations before you finally get to the bottom of expression. And the way it did so is actually pretty interesting. It's actually evolved here 2 repressor binding sites. And you can see, based on the fact that these many of these bases are red, if you screw up binding of this repressor, it's actually going to go right back to the top of expression. So it took many mutations to get down here, and now it takes 1 mutation to get back up. So very different evolvability characteristics for these sequences. So that brings me to the conclusion of this part. Many mutations can alter expression, and expression changes for at least single base pair changes follow a power law. Most promoter sequences have accessible large effect mutations, and very few mutations are required to go between the expression extremes, either from low to high or high to low. In most cases, as we saw with UTH1 at the end, it's not always true. And satisfying competing objectives is much harder than a single objective. So we use this oracle to identify expression evolution using these population genetics data. I found a really interesting example, CDC36, where there was convergent expression adaptation in both the expression level and in the TF that actually mediates that expression level. And 3 archetypes explain the evolvability of most promoter sequences. And we can see in this archetype space both the evolutionary history and future of the promoters, because if a promoter is on the outside, it will evolve diverse phenotypes very easily. And if it's stuck near the robust cleft, then it will be fixed. So that brings me to one of the things we're working on now. So Rafi is a graduate student in the lab, and in collaboration with DREAM, we're organizing this DREAM challenge to see who can make the best gene expression model, essentially. So we have an unpublished data set where we did the same approach of sorting yeast, random sequences, and sequencing them to measure expression. And we're trying to see if we can make a better model than the one Ishit made. He made a very good model, but he's only one person, and I don't think one person can solve this problem by himself. So here are the acknowledgements. I acknowledge the people as I went through, so I'll just leave that there. And I also wanted to paste in the chat. You can use the QR code if you want, but there's a link to the competition page if you're interested in joining. And any more questions, I'd be happy to take now.
Thank you so much. I think there is at least we can start with that. There's a question from the previous part of the talk from Alexander Morin. So it's about how in the 2020 ENCODE release, they were looking at the accessibility focused chromatin papers, and they put a general framework of regulatory elements wherein 200 base pairs have about 5 to 6 transcription factors, where the transcription factors tend to be slightly more spaced than being directly overlapping. Does that align with your insights of what you see in yeast?
Yeah, so if I understand the question correctly, the person is referring to a paper that's based on DNA's hypersensitivity, so footprinting of transcription factors. And there you would only see transcription factors that are bound very robustly. All these very low transient effects where transcription factor is just binding for a second and then floating away, you wouldn't see it in the DHS data. So in that sense, it is consistent, but their data doesn't lend any support for these low affinity, weak interactions actually doing something.
Cool. We have a question from Philip Richmond. In your multi-strain yeast model, how much do you think differences in transcription factor sequence, for example, amino acids in DBDs, interact with differences in promoter sequences? An example is RIM101 in the AQY2 promoter. Is this something that you could incorporate into the model?
Yeah, that's a really great question. So to restate, sounds like Philip is asking about how genetic variation in the protein sequence of the transcription factor will affect its function and its ability to read DNA. We're actually exploring this now using a modified version of this system, but it's a great question and we don't really know the answer. Philip points out Martha Bullock's work, so that's like the current, as far as I know, best knowledge in that space.
So, and by the way, if people would rather speak up, their like talk and like raise their hands or just unmute themselves to ask the questions, please go ahead. But I guess we do have one more question from Girman. Scalability aside, did you see any pros of using transformers on the task, on this task compared to CNN models?
Well, we hope to explore this more in the DREAM competition, but at least the models we considered, the transformer model did appear to predict better than the CNN. So it had fewer parameters and it predicted better. And presumably, if it has fewer parameters, it can also converge to a better optimum, although I'm not 100% sure about that because that assumes some other things, but that's the way I am thinking about it.
Thank you. Well, there's another question from Eric. Thanks for the great presentation. The biochemical model showed that there are many transcription factors binding, each with very small effects, where the deep learning model showed that a few mutations can induce large changes in expression levels. Do these findings contradict each other?
No, I would say they don't contradict each other. So just sharing this slide again. So this is showing what the deep learning model is predicting for all possible mutations to yeast promoter sequences, what their, this is the absolute value of the effect size. And so what this is saying is that there are some that have a very large effect size, but there's also many other mutations throughout the sequence that are affecting expression, just not always in this very large way. And biochemically, this makes sense. There are some transcription factor binding sites that are going to be very important in the expression of that sequence. So in the slide before, these are like the greater than two-fold interactions between transcription factors and the sequences they regulate. So this is pretty consistent between the two models.
Yeah, thanks so much. I guess we'll take this question and maybe one more question if there's one last question. From Fiel, would you say that evolvability archetypes are analogous at all to certain models used in phylogenetic comparative methods? The Ornstein-Oehlenbeck model comes to mind, for example.
Well, they're definitely related. Is it analogous? Yeah, I don't really think it's analogous, but they are definitely related. And like the Orstein-Uhlenbeck model is capturing how expression, it's actually a lot like this, what we did here. So how expression changes as the sequences diverge. So this is like captured indirectly in the space, but one thing that is captured in this archetype space is like the rate of diffusion essentially of these sequences, and the ones near the middle basically don't seem to be able to evolve any further. They're stuck in that space, or it would just take a very large number of mutations to get out of the space.
Okay, thanks. And I guess our last question for today, again from Philip. In your model, is it possible to incorporate known relationships between transcription factors, such as protein-protein interaction, data from yeast to hybrid, etc.? Do you think this would improve the model or just confound it?
Well, that's a great question too. So it probably wouldn't confound the model, because worst case scenario, the model is going to say your known interaction doesn't actually do anything here. So you would tell it about it and it would throw it out instead of paying attention. So you could use known interactions. The problem is that most possible interactions were never even explored. So you can't very easily, you can't just tell it about all of the interactions because we don't know all the interactions. So in my mind, that kind of data, the yeast-2 hybrid data with like transcription factors interacting and UC type halase group also has a great paper showing TF-TF interactions. I think we should use that as validation of the parameters learned, but you could definitely, like if we found some way to do it for all by all in a robust way, and we could just give those to the model, I think that would be a better way of, like, you would probably learn a better model in the process.
Well, thank you so much. I really want to thank everybody for attending, and I especially, of course, want to thank our two speakers today, Brad and Dr. Carl. And I hope to see you all next month for the career panel, and definitely next season back in person. So thank you so much, everybody. Have a good day.